I'm going to talk about breast cancer-related hormonal alterations by COREPRESSOR and COR1. So there are many studies between vitamin D and subtypes of breast cancer, especially TNBC, where there was a growth factor reduction after administration of active form of vitamin D. Vitamin D has important functions at cellular differentiations, apoptosis, and telemodulation cycle. Co-repressors have been related with vitamin D and recruit activity of stones deacetylase silencing genes. And COR1 is the co-repressor we're going to study about. Estrogen and NCRR1 has been related to in many studies where we have the tamoxifen-resistant breast cancer, where we have the upregulation of coactivators and downregulation of co-repressors. CO2 is a molecular where is upregulated by estrogen and it generates a process of ubiquitination of NCRR1. So when we have the upregulation of CO2, we have the downregulation of NCRR1. So the methodology is by RNA extraction, cDNA synthesis, and real-time PCR, where we use those kits we have on the screen. So the results, we have the expression results, where we have the reference gene beta actin. The samples were 40 samples, being 6 TNBC, 10 HER2 positive, 22 luminals, and 2 inconclusive and 3 controls. The CTs were between 18 and 24. So to make the graphic, we use the Prism software where we use the log2 expression. Above 1, we have the upregulation, and below 1, we have downregulation. In NCR1, we had the expression of all TNBC patients, where 2 had high expression of NCR1, while 4 were downregulated. 11 luminals had high expression, while 11 were downregulated. Of 10 HER2, 6 expressed it, 3 being upregulated and 3 being down. In ESR2, all the luminals patients expressed estrogen receptor. 2 NBC patients had a low expression of ESR2. Of 10 HER2 positive, 3 had low expression of, and 1 inconclusive were expressed up. VDR were basically downregulated in all subtypes, only 2 TNBC expressed, 15 of 22 luminals expressed, and 6 of 10 HER2 positive expressed. CA2 were also downregulated, with 20 luminals expressing, 6 HER2 positive expressed, 3 TNBC expressed, and 2 inconclusive expressed. So to conclude it, the NCOR1 can be an important molecular in the regulation of key pathways of breast cancer. The low expression of VDR together with low expression of NCOR1 in TNBC patients may indicate a downregulation of vitamin D receptor, favoring the cancer development. Further analysis with healthy women should be performed, and ESR2 and CO2 levels were decreased. We have the NCOR1 elevated in many cases. So it is important to study in healthy women and should be performed a further analysis.